Hello students, welcome to EPG Pathshala. I am Mohan Chand Joshi, Assistant Professor from Jamia Millia Islamia. Today we are going to talk on a module, Plant Transformation Vectors, Ti, Ri Plasmid, under the paper Genetic Engineering and Recombinant DNA Technology, Part 1. After completing this module, you should be able to understand what are Ti and Ri plasmids are, and how agrobacterium mediated transfer of tDNA into plant's genome happens, what are the differences and the similarity between Ti and Ri plasmids are. Agrobacterium. So Agrobacterium is a gram-negative bacteria which belongs to a family of Rhizobiaceae. It grows aerobically, that it means it can grow in a condition where the oxygen is present without forming endospores. It is a rod-shaped bacteria which contain 1 to 6 flagella, as you can see in this electromicrograph of 1988, that this is a agrobacterium has 1 to 6, almost 4 or 5 flagella. There are different disease causing bacterium species, agrobacterium species, and here is the list of them. So Agrobacterium Tumefaciens causes crown gall disease. Agrobacterium ruby causes small galls. Agrobacterium Rhizogenes causes hairy root disease, and Agrobacterium Radiobacter species is avirulent, that means it does not cause any virulence into the host. Agrobacterium Tumefaciens is now renamed as Rhizobium Radiobacter. It is a rod-shaped gram-negative bacteria and it contains a Ti type plasmid. This bacterial species can infect dicots and cause tumor, and dicotyledons such as walnuts, tomatoes, roses are its prime target. It can cause tumor-like growth by transferring the Ti plasmid, which is present in as an extra chromosomal copy, into the plant cells, and could result in a uncontrolled growth of plant cells, which forms a tumor. Here is an example of how Radiobacter causes tumor in plant cells by transferring the Ti plasmid. So on the left hand side you have a healthy plasmid, and this is a soil bacteria. So this bacterium is present in the soil, but it's not been able to infect the plant until plant sustains an injury. Now once plant sustains an injury, this bacteria can go to that site which is damaged by using chemotaxis method. I will explain that in detail in next few slides. And once it goes to that infected site which is wounded, the agrobacterium can colonize there and transfer the Ti plasmid and can result in the formation of a tumor, which is shown in the picture here. So soil bacterium, which is non-harmful, can turn into a tumor causing bacteria. Agrobacterium Rhizogenes, that is now renamed as Rhizobium Rhizogenes. This is a rod-shaped bacteria, gram-negative soil bacterium present in the soil. It harbors Ri type plasmid, and this Ri type plasmid can induce infection to the root site and causes hairy roots if this plasmid is transferred into the plant cell. Rhizobium Rhizogenes induces hairy root via Ri plasmid. As I explained in the previous slide, the rhizogenes contain Ri plasmid, and in this you can see a healthy plant. In the soil you have this bacteria present, rhizogenes is present, and if the site is wounded, this bacteria can recognize this wounded site by chemotaxis, can go there, infect the plant, and results in uncontrolled growth, resulting in hairy roots, as you can see in this picture. Ti plasmid. These are naturally occurring plasmid inside the agrobacterium, and their size is huge. They are almost 200 kb in length and contains major genes such as tDNA. And tDNA is a stretch of DNA which is bordered by 2 repeat sequences, TL and TR repeats as they say it, and also contain Virulence Operon. And this Virulence Operon contains genes where A, where B, where C, where D, where E, and where G. And each of these genes encode for a protein that is required for a stepwise transfer of tDNA into plant cell. Second is the opine catabolism gene. Ti plasmid has a capability to catabolize various opine such as octopine, napoline, succinopine, and leucinopine. These and Ti plasmid have origin of replication, which allows its duplication inside the agrobacterium. And lastly, it also has a conjugal transfer gene, Tra, which allows its transfer from agrobacterium into plant cell. Here is a genetic map of a Ti plasmid. As I explained in the previous slide, Ti plasmid contains an origin of replication which allows the duplication of this plasmid. They are almost 200 kb in size, very large. It also contains a virulence region which is shown in the right hand side, and also contain genes that can catabolize opine, a chemical or molecule which is secreted by plants upon its infection, and also contains genes which are involved in conjugal transfer of this particular plasmid into the plant, and also contains tDNA. tDNA is also named as transfer DNA, and if you zoom into tDNA, what you see that there are genes in it which are auxin genes, cytokinin genes, and opine synthesis genes which are present in tDNA, and these genes are bordered by repeats elements, left repeat as well as right repeats, and these repeats are crucial for the transfer of this plasmid. In particularly, right repeats are very important for the successful transfer of tDNA into plant cell. Ri plasmid. Similar to Ti plasmid, Ri plasmids are also naturally occurring plasmid inside agrobacterium, but their size is huge. They are even bigger than the Ti plasmid. Their size ranges from 200 to 800 kilobase pair. They also contain crucial genes like Ti plasmid, but instead of having one tDNA, they can have maybe one or more or two DNA sequences. And if they have two tDNA sequences or regions, then they are named as tLDNA and tRDNA. They contain Virulence Operon gene just like Ti DNA. They also have genes which are involved in opine catabolism, but they are different from what we see in Ti plasmid, and the opine that they can catabolize is agropine and menopine. Ri plasmid also contains origin of replication that allows its replication inside the agrobacterium, and also has a gene for conjugal transfer. Here is the genetic map of a Ri plasmid, and as I mentioned in previous slide, it contains origin of replication which allows it to replicate inside the agrobacterium. It also contains opine catabolism gene, conjugal transfer gene, virulence region. All these regions are similar to what we see in Ti plasmid. However, the tDNA here looks very different, as you can see in the schematic also. It contains tDNA repeats for sure, left and right repeats, but it may contain up to 2 copies of tDNA. Therefore, now we say them tRDNA, that is right, T left DNA, and also has a space between these two is called tcDNA. Ri plasmids are bigger in size compared to the Ti plasmid size. Virulence Operon. This region is present in both Ti plasmid and Ri plasmid, as I mentioned earlier. It contains a range of genes which are involved in the transfer of tDNA from bacteria to the plant cell. Genes in the virulence regions are grouped into operon called Vir A, B, C, D, E, F, G, and that codes for enzymes responsible for tDNA transfer. To start with Vir A gene, it codes for a receptor which reacts to the phenolic compound that are released by a wounded plant. This could be acetosyringone, syringoaldehyde, or acetovenilone. Now this gets autophosphorylated in presence of these phenolic compound. As it works in tandem with the Vir-G as a 2-component system, it can also phosphorylate Vir-G, and then this Vir-G gene activates expression of other virulence gene after binding to a consensus sequence of Vir-A that is phosphorylated. Now Vir-D D1 and D2. These two proteins are endonuclease enzymes which targets the repeat sequence of tDNA border and can induce a damage only on one strand, chews it up, and resulting in a 5-strand end of a single strand DNA. So these endonucleases will go and specifically bind to the tDNA, will chew up only one strand of DNA and results in a single stranded DNA. And that DNA, the single stranded DNA, is protected by the binding of Vir-E gene, which prevents it from any nuclease attack and intercalates with lipids to form channel into the plant membrane. There are two proteins, Vir-E1 and E2, both act as a chaperone to protect this particular tDNA, single stranded tDNA, against endonuclease activity. Now the last is Vir-B gene. As we have a tDNA complex which is formed by the binding of Vir-E chaperones and the proteins to the single strand DNA, Vir-B is a protein which forms channel, which allows the transport of these tDNA complex from bacterial cell membrane into the plant cell. So it can form a channel through bacteria into the plant cell. tDNA sequence, or as we say, transfer DNA sequences. The tDNA sequence is present in both Ti and Ri plasmid. In Ri, however, there are two tDNA sequences, but the genetic map for tDNA remains the same. tDNA or transfer DNA is almost 24 kb long, and out of the entire Ti or Ri plasmid, it's only the tDNA sequence that gets integrated into the plant genome. Apart from this tDNA sequence, all the genes which are present in Ti or Ri plasmid are there to facilitate the transfer of this tDNA into the plant cells. In tDNA, at each end of the tDNA, you have two border sequences which are 25 base pair repeat and are crucial for tDNA transfer. The right end, specifically the right end repeat, is very important for the tDNA transfer, because if you remove the left end repeat sequence from tDNA, bacteria is still able to transfer the tDNA into the plant cell. tDNA contains important genes which are required, or which synthesize or which encodes for opine and phytohormones. These two genes or these two products are important. Opine is important, which is derivative of amino acid for bacteria as a source of carbon and energy. Phytohormones such as cytokinin and auxins are responsible for uncontrolled growth in the plant cell, which leads to the tumor formation. Here is a schematic overview of how Agrobacterium infects the plant cell and transfer its tiDNA or Ri DNA into the host cell. So the very first step that I mentioned in earlier slides also, that if a plant gets wounded, so what happens in that case, if plant gets a wound, it releases a phenolic compound which is acetosyringione, is shown in this picture here. So this particular compound, phenolic compounds, is transported into the bacterial cell by Vir-A gene, which results in the step 3, leading to the autophosphorylation of Vir-A protein. Vir-A autophosphorylation activates the expression of Vir genes and leads to activation of a range of virulence gene, including D1 and D2. Now D1 and D2 will specifically excise out the tDNA molecules from the tiRNA and will make a single strand tDNA. As you can see in this picture, you have Agrobacterium with a Ti plasmid, in which the Ti plasmid, the tDNA sequence is nicked by Vir-D1 and D2 gene, and a single strand is chewed up, and we have a single tDNA strand. Once the single strand tDNA is formed, it needs to be protected against the endonuclease or exonuclease that are present in the cell. Therefore, immediately this protein complex is bound by a chaperone or a protein Vir-E1, which makes a complex and is named as T complex. Now this T complex is much more stable, and it can form a small, it can make a filament like structure which is 2 nanometer in its width, which is capable of passing through the membranes of bacteria as well as a plant. But in order to do that, it requires an assistant from other protein, which is a Vir-B protein. Now this Vir-B protein forms a channel which allows the transport of tDNA complex from bacteria direct into the plant cell. And as you can see in this schematics, you have a tDNA complex shown in a filament here, where single strand DNA shown in red is captured or protected by a blue protein, which is represented here is Vir-E2. Now this complex need to be transported, the tDNA complex need to be transported from Agrobacterium into plant cell, and in this picture here we can see a plant cell wall, we can see plant nucleus and a plant genome, and Vir-B will allow a transport of, will form a channel which allows these 2 nanometer filament complex to translocate from bacteria via plant cell into the plant cytosol. In the next step, once the tDNA is into the bacterial cytosol, it will get into the plasmid, into the cell nucleus as well, and now the protein which are protecting the single strand DNA need to be cleaved off, so they are removed, and after that the tDNA, the single stranded DNA, get integrated into plant genome. However, the mechanistic understanding of this integration as well as transport into the nucleus remains uncharacterized. The mechanism which is underlying this transport from cytosol into nucleus into the bacterial genome remains unknown and needs further characterization. So once the tDNA is integrated into the plant genome, and as you remember, the tDNA contains our genes of auxin, cytokinin as well as opine. Now these can utilize the plant transcription machinery and will start its own transcription as well as expression. And as these auxin and cytokinin genes are synthesized within the cell, it results in uncontrolled cellular growth, and which cannot be stopped, and that results in the formation of tumor. As you can see in this picture, the red tDNA is integrated into the plant genome, and the synthesis of these phytohormones results in the formation of a tumor. So what are the differences between Ti type plasmid and the Ri type plasmid? As we know, the bacterium species which harbors these Ti and Ri plasmids are different. Apart from that, there are characteristics of these two plasmid which differs, and as you can see in these tables, the characteristics are mentioned on the left side. Then you have Ti plasmid and Ri plasmid. First and foremost difference, which is very obvious between these two plasmid, is the tDNA. In Ti plasmid, we have one copy of tDNA, but in Ri plasmids, we can have two copies of, either one or two copies of tDNA, which makes Ri plasmid bigger in size compared to the Ti plasmid also. The opine catabolizing gene. Both Ti plasmid and Ri plasmid have a different set of opine catabolizing genes. In Ti plasmid, we have octopine, nopalene, succinomapine, succinoopine, and leucinopine. In Ri plasmid, we have agropine type or manopine type. In both these Ti and Ri plasmid also have similarities, that both of them contain only one copy of virulence genes. Ti and Ri plasmid both are virulent to the plasmid, to the plant cell, and Ti plasmid cause tumor, which is also called as crown gall, and Ri plasmid cause hairy root disease. And as I mentioned earlier also, they both reside, the host of these two plasmids are different bacterial species. Ti plasmid is found in Agrobacterium Tumefaciens, which is now named as Rhizobium Radiobacter, and the Ri plasmid resides in Rhizobium Rhizogenes. So students, let's now summarize what we have learned in this module. First, that Ti and Ri plasmids are present in a naturally occurring two different bacterial species, Agrobacterium, and that Agrobacterium is a soil bacteria, it's a gram-negative rod-shaped bacteria. Both plasmids contain tDNA, that's a transfer DNA sequence, and the virulence region. The virulence region contains a range of genes, virulence gene, which are responsible for the transfer. Compared to Ti plasmid, Ri type plasmids are slightly bigger. their size range from 200 to 800 kilobase pair, and they contain up to 2 tDNA sequences also. So how Agrobacterium recognizes the wounded site and causes the infection or causes the tumor? So what happens once the plant gets injured? It releases a phenolic compound, and these phenolic compounds are recognized by virulence A gene in the plant of the bacterium, which get autophosphorylated. It transfers the phosphate group to virulence gene G, which results in the downstream expression of other virulence genes. Now, once the genes are expressed, virulence genes are expressed, they excise the tDNA, the double-strand DNA, into a single-stranded DNA, which then binds to several to the different virulence gene A, which protects the single-strand DNA against against the endonuclease of the bacteria, so that the T-strand DNA is not chewed up. Once the tDNA is bound with the Vir E genes, it translocates with the help of Vir B gene into from bacteria cell membrane to the plant cell membrane, where it is released into the cytosol. It travels from the cytosol to the host, to the plant nucleus, and gets integrated into the plant genome. However, how this transfer between cytosol to nucleus and from nucleus to integration into the plant genome remains uncharacterized. But once it gets integrated into the plant genome, tDNA utilizes the host transcription factor and synthesizes and expresses auxin, cytokinins, and opine genes. And opine is used by bacteria for their survival inside the host. But expression of auxin and cytokines results in uncontrolled differentiation of plant cell, which results in the crown gall or hairy root disease. So students, by now you will have learned the mechanism through which Ti and Ri plasmid from Agrobacterium causes these disease in plant cells. Thank you.